Students, in this lecture, I'm going to talk about adaptation of bacteria to cold stress. This is paper BCT 205. So, bacteria, you know that they grow at different temperatures. They grow at low temperature. You have examples of bacteria which can grow at low temperature, high temperature, and normal temperature. The low temperature are called as which can adapt and grow to nicely to the low temperature are called as the psychrophills. The bacteria which has a growth optima at normal temperature, that is ranging from 25 to 35 degrees Celsius, are mesophils, which are growing at high temperature, which have growth optima at high temperature, are called as thermophiles. So, adaptation of these different kinds of bacteria to cold stress, or when they are put into temperature 15 degrees and below, how they behave is almost similar. Like if you take an example of a mesophil bacteria like E. coli and Bacillus subtilis, how they adjust, there is some overlap with this mechanism with above mesophil temperature like around 60 degrees. Still there you can see the similar pattern of arrangement, adaptation to the cold stress. So the bacteria which have optima at very low conditions, they will have normal metabolism happening, and there is hardly any adjustment at the cold temperature. But mesophils, they show lot of changes when they are put into low temperature, so they adapt constantly to these new environment. So apart from cold stress, they can have any other kind of challenge, like reduction in nutrients, or oxygen availability, or osmotic stress, or drastic changes in temperature like high temperature. So all these changes, the microorganisms are constantly adapting. So if there is a sudden drop in temperature, you're growing the E. coli or Bacillus in at 37 degrees, suddenly you reduce the temperature to 15 degrees. There is number of important changes that occur in cellular physiology, like there will be decrease in membrane fluidity and stabilization of secondary structures of nucleic acid, which may lead to a reduction in the efficiency of transcription, translation, and degradation. So, cold shock response enables these microorganisms to counteract these unfavorable conditions created by setting up the organisms to the low temperature. In laboratory condition, the cold shock response is triggered by just bringing down the cultural condition, that is optimal temperatures, to lower temperature like 15 degrees. So upon this shift, so now the bacteria has an exponential phase at 37 degrees Celsius, and suddenly you shift the organisms to low temperature. There is kind of adjustment, or there is transient halt of the cell growth for 3 to 6 hours period, and this period is termed as acclimation phase. In this acclimation phase, there is production of lot of proteins, which normal production of protein stops, but the cold inducible production of proteins are becomes more and more. The acclimation After this acclimation phase, cells become adapted to lower temperature, and again they resume their growth, but comparatively they will be at a slower rate. This The expression of this cold inducible protein declines the bulk protein synthesis. So there is lot of changes at this acclimation phase and their growth phase. So there is adaptation at the cell membrane site, adaptation in RNA metabolism like transcription, degradation or stabilization or translation. There is lot of changes happening, and one mechanism we'll see of cold inducible specific proteins in under cold conditions. So, if there is a rapid temperature downshift that can induce the phase separation of cell membrane phospholipids, because of which there is constantly decrease in fluidity of the membrane and increase in permeability. For example, if you take gram-negative bacteria, they are composed of lipopolysaccharide, which has a distal O antigen, and a core polysaccharide, that is lipid A. The E. coli lipid A consists of two glucosamines which are attached with attached acyl chains, the fatty acids. Those fatty acids are laurate, usually detected in the cells, which is seen at 37 degrees. But when you subject this E. coli to low temperature, there is decrease in laurate and there is increase in palmitoylate. So this increases the membrane fluidity and lowers its phase transition temperature. Okay, counteracting the effect of low temperature. So because of this sudden cold, there are cold induced acyl transferases which are responsible for attaching palmitoylate to lipid A molecule upon temperature downward shift. In case of Bacillus subtilis, the adaptation of membrane fluidity involves rapid desaturation of fatty acids in already existing phospholipids. So this happens by induction of fatty acid desaturases regulated by the sensors, that is DESK, and response regulator DESR. So DESK has a transmembrane domain, and that is the sensor of membrane fluidity. So at low temperature, causes a decrease in membrane fluidity, which favors the active site of DSK kinase. DSK phosphorylates the transcription activator DSR, which subsequently binds to the promoter of DS gene and activates D5 desaturase. So, this enzyme introduces a double bond into pre existing fatty acid tail of phospholipids inside the cell membrane. So that helps in increasing the membrane fluidity. Adaptation to RNA metabolism. So most of the cold inducible proteins are involved in RNA metabolism. There are like many proteins. These proteins most strongly induced are nucleic acid chaperone form of family of cold shock protein termed as CSPs. Uh, they are very crucial in RNA metabolism, hence by helping in adaptation. Low temperature causes stabilization of secondary structures of nucleic acid, and this can impair transcription, RNA degradation, and translation. The main function of this cold induced proteins involved in RNA metabolism is to prevent the secondary structure formation or to facilitate degradation of structured RNA. Proteins from CSP family are nucleic acid chaperones, and this activity prevents the formation of secondary structures and facilitate their degradation by cold shock exoribonuclease. One of the exoribonuclease are PNPase and RNase R. So RNase R is a 3' to 5' exonuclease in E. coli that efficiently degrades double stranded RNA. Uh, when you have this low temperature, the curvature of the DNA changes and it becomes more and more negatively supercoiled. So, this effect partially depends on the activity of histone like protein, that is HUPB and gyrase, an enzyme capable of introducing negative supercoil. The effect of this negative supercoil uh, uh, in turn trigger the transcription of cold induced genes in a way that and that has to be like still clearly need to be understood. So the majority of this cold induced genes are the proteins what I mentioned, cold specific proteins, CSP family of proteins. They also bind to DNA and bring about help in the transcription. Apart from that, they help in maintenance of chromosomal structure. So if you express this kind of proteins in plants, they help the chromosome folding. One or two of the important proteins are CSPA and CSPE, which are shown to involve in transcription at low temperature, acting as transcriptional anti-terminators, preventing hairpin formation that may lead to premature termination. So, one more example of anti-terminator proteins which are expressed under cold stress is NUSA. So, there is one more transcription factor, the RPOS. It's a sigma transcription factor. Sigma S is um, not induced is not a cold induced protein, but it has been connected with expression of genes in response to many stress, including the shift to low temperature. So in E. coli study there are like more than 40% of the gene preferentially expressed at 23 degrees Celsius, which are shown to control RPOS gene. Several proteins from the cold shock protein family are involved in RNA stabilization and or degradation. The chaperone functions of these cold shock proteins are very crucial for mRNA stability in cold condition. CSPA, the major cold induced chaperone protein, destabilizes the secondary structures and functions as a chaperone to maintain RNA in a single stranded state, which favors the degradation. CSPE acts in opposite way, that is hampering RNA degradation. CSPE binds to poly-A tail, interfering with their digestion by PNPase, and can also inhibit RNA internal cleavage by RNase E. There is one more protein called as dead box helicase protein, which is induced under cold condition. Uh, This is a protein which is mainly incorporated into the RNA degrading machine, that is degradosome. It has a helicase activity and it helps to degrade structured RNA. So this dead box helicase associates itself with RNase E, which is the main endonuclease, and they form the scaffold of the degradosome complex. So the helicase activity of this uh, dead box helicase protein is also involved in ribosome biogenesis in low temperature condition. PNPase is one of the main E. coli exonuclease which increases its level like more than twofold when suddenly the cells are dropped into low temperature. It helps to repress the production of cold shock protein family of protein at the end of assimilation phase. So, so indicates that these proteins are contributing to specific degradation of cold shock protein mRNAs. So three important exonucleases are there, that is PNPase, RNase 2 and RNase R. So RNase R suppresses the dead deletion phenotype at low temperature. That is, RNase R is a cold shock protein, increasing its level about tenfold under cold conditions. That means they are like they helping digest RNA secondary structures without the help of helicases. They help unwind secondary structures um, which are based and help in degradation of RNA at low temperature. So why this degradation of RNA is happening so much? Because only the proteins which have to be favored for um, adjusting to cold temperature has to be produced. The other proteins which are like transcribed should not be expressed more, that is translated more. So more and more of these exonucleases are synthesized, and then they regulate, they bring down the energy status only towards the synthesis of the stabilizing proteins. So as a result of this sudden bringing down, there is a translation block with like only the cold shock inducible proteins are produced. PY is one of the proteins which is involved in this block in translation. PY, what it does is it reduces translation of some mRNAs due to its capacity to bind to ribosomal 30S subunit. So when this PY protein associates with ribosome, um, it can modulate the function of the ribosome. So at low temperature, it can cause misfolding of the RNA, which is not required. There are other proteins like initiation factors 2, initiation factor 3, which are also modulated under the effect of the cold stress. So there is another proteins which has a role in this translation of protein folding, which is a trigger factor. This trigger factor level suddenly increases in cells which are dropped or which are kept under cold conditions. Um, this trigger factor is thought to help protein synthesis and help the folding to continue in the cold condition, and also to maintain the pre-existing protein in functional form by promoting refolding of cold damaged protein. So it's like it is involved in protein folding at a low temperature. So we'll take one example. During adaptation to cold, expression of most proteins is downregulated, but cold induced proteins are still expressed or even induced. So there is, if you see, generally there is no transcription factors responsible for this induction, and regulation, uh, majority of the regulation lies in the changes in the mRNA stability and accessibility to translation machinery. Cold shock protein A could induce cold induction, has been most extensively studied, and it is the one which is highly regulated under cold conditions. A cold shock protein A promoter responds to temperature downshift. The core promoter of cold shock protein can be induced by cold shock up to threefold at 15 degrees Celsius. Cold shock protein RNA levels are also increased due to stabilization of the transcript. So the mRNA, cold shock protein A mRNA, is extremely unstable at 37 degrees but highly stable at 15 degrees with a half-life of 12 seconds in 37 degrees and 15 minutes at 15 degrees. So at the temperature shift from 15 to 37 degrees, most of the cold shock RNAs are degraded. These changes in stability are thought to be caused by the important 5' untranslated regions. They show some structural rearrangements which cause the stability at 15 degrees Celsius, but instability at 37 degrees Celsius. So, structural changes of this cold shock protein A 5' untranslated region impact the messenger RNA by opening the translation initiation region at low temperature, but not at the higher temperature. So, other initiation factors like translation initiation factor 3, initiation factor 1, together they act as a transfactor to facilitate the cold shock protein A mRNA production. So, these three together act and cold shock protein production. No, regulation. Cold shock protein A production also one of the regulatory mechanism. So there are many different proteins which are induced under cold shock. That is like pyruvate dehydrogenase decarboxylase is expressed more. Cold shock protein A is expressed more, which is an RNA chaperone and anti-terminator transcription enhancer, and homologs of CSPA, that is CSPB, E, G, are produced. They are also cold shock protein. They show like they are also transcriptional anti-terminators, and dead box protein D, DNA A, gyrase is produced more. There are more nucleoid proteins which are produced. DNA K chaperones are produced, and a lot of transcriptional terminators and anti-terminators or elongation L factors are produced. Exonucleases PNP, RNR are produced. Ribosome binding factors RBFA are produced, REC are produced. These are all induced under cold stress. So protein Y, which is associated with 30S ribosomal subunit and inhibits translation, is also produced excessively under cold stress. And in cold stress, these are the minimal change. There are a lot of other changes which are happening. Minimally, these are the changes which are observed clearly in many bacterial species under cold stress.